Okay. I want to thank Spila and the organizers for inviting me and giving me this opportunity to present our research. I'm very happy to be joining, even remotely, joining from Boston. Good afternoon to everyone there. Also, thanks, Anna. That was a great overview of the therapeutic approaches. So I'd like to start with the slides. Am I controlling them or are you?
You can share the slides if you want.
Ah, okay.
Yeah, if it's not possible, we can do it for you.
Okay.
Did you send the slides?
I did send them. Let's see.
Hi, Michelle, Isana. If there is a way that you can share a screen and you are in full control to passing the slides and anything
Is
you
it
need.
visible now?
Yes, perfect. Thank you.
Great. Okay. All right.
Thank you.
So our talk is the small molecule efficacious therapeutic treatment for CTNNB1 syndrome. Okay, let's see it. Okay. For some reason, it's not advancing. Let me go out and try this again. I'm going to go in again because it wasn't advancing. Okay. It won't advance. I think I'm going to need you to show them.
Okay. You need to press one time on the screen. Try to tap there. Anna will be helping.
So, Michelle, the technician is telling me that once you share the screen, you have to first click one time, like enter or with the mouse, one time on the screen. Normally,
Okay,
they should
let's
get
try it
started.
again.
Let's try again, and otherwise, we will try from here.
Okay, that's working. Okay, so let's go back. Okay, so here's our title. So as you're all well aware, CTNNB1 syndrome is caused by sporadic mutations. These are heterozygous loss of function pathogenic variants in CTNNB1, and different patients have different mutations that span the gene. And you've heard that there are two different alleles of CTNNB1. So one is mutated and essentially isn't expressed properly. So you have half of the amount of the protein generated from that gene. That protein is beta-catenin. So the core molecular pathology is insufficient levels of beta-catenin. And it's really well known that this is a high confidence risk gene in humans for intellectual disability and autism spectrum disorder from work of many groups. It's also been recently shown that pathogenic variants in this gene are a lead genetic cause in individuals misdiagnosed with cerebral palsy. And in addition to CTNNB1, there are several other human genes that link to intellectual disability and autism, and they're predicted to cause reduced levels of beta-catenin or reduced function. So it's a very important disease pathway. And our work has been focusing on finding treatments for CTNNB1 syndrome. We work closely, we're a lead researcher in CTNNB1 Connect and Cure, the patient advocacy group in the US. We're on the scientific advisory board of CTNNB1 Italia. We closely collaborate with medicinal chemists at the Broad Institute, associated with Harvard and MIT, and they develop the small molecules that I'll be telling you about today. And based on our work with the Broad on these small molecules, we have a research agreement with the National Institute of Health and CATS, the National Center for Advancing Translation Scientists. It's their therapeutic development branch. And we've received a prestigious award from the Oxford Harrington Institute as a rare disease scholar, and we interact with these fantastic advisors that have decades of experience in pharma and bringing therapeutics to clinical trials. So the biggest problem with CTNNB1 syndrome is right now there are no treatment options. And you can hear from all the talks over the conference that many groups now are focused on addressing the gaps. The gaps are limited knowledge of the underlying pathophysiological changes. And until recently, we had few preclinical models. So we're taking different approaches to address this. My lab has developed an in vivo mouse model, a CTNNB1 heterozygote, full body heterozygote. Only one allele of CTNNB1 is expressed. And we most recently have developed different human cell models. The ones I'm most excited about that I'll be sharing with you today are actually patient-derived cells. And from our work, we're getting new insights into molecular and functional changes and identifying an efficacious, safe therapeutic treatment. I'll focus today almost solely on the small molecule work, but my last slide is on a novel RNA-based therapeutic that we've developed in collaboration with Johns Hopkins. And we do have a patent with the Broad Institute for GSK3 inhibitors as a method of treatment for CTNNB1 syndrome. That's the data I'll be sharing. So you've heard from various talks about CTNNB1 and the core problem, insufficient levels of beta-catenin. Beta-catenin is expressed in every cell of the body. It's important at every stage of development, from the very beginnings of life until the end. And its function varies with cell types and its position within the cell. I'm going to focus mostly on nerve cells here because that's really one of the core features of CTNNB1 syndrome problems. So it has two major pathways that are essential for the proper maturation and function and plasticity in the brain. So at the synaptic connections, this is a site where one nerve cell contacts a target. This is rapid information transfer. It's important for all functions of your body, all behaviors, everything you do. So there are adhesion molecules. You've heard about the N-cadherin. Beta-catenin links to N-cadherin, and together they will play a role in stabilizing synapses, controlling the number of synapses you have, and how synapses get larger with learning and memory. So very important protein complex at the synapse. Beta-catenin will also go into the nucleus, as you've heard, and it controls Wnt-dependent gene expression. Wnt controls cell proliferation, cell fate, axon guidance. It also is important later in development plasticity for learning and memory. So these are critical roles in the body. And in that Wnt pathway, what happens is you have this opportunity to control your beta-catenin levels. So typically, you are keeping, every one of us keeps beta-catenin within a certain level of expression. And during activity in the brain, there's a soluble molecule released, a Wnt, that acts with receptors on the surface of a nerve cell or a target like a muscle cell, and it activates a pathway that actually leads to increases in beta-catenin levels. And it's centered on this protein, GSK3, glycogen synthase kinase. Like beta-catenin, GSK3 is expressed in every cell of the body, and it functions normally. It's an enzyme. It's always active. It causes a change in the protein beta-catenin. It's a post-translational modification of phosphorylation, and that causes beta-catenin, when phosphorylated, to bind to other proteins that leads to its degradation. But the cells during activity inhibit that enzyme, transiently inhibit it, and that prevents the phosphorylation. So the beta-catenin accumulates, and it can now go back to the synapse and interact with cadherins, and it can go into the nucleus and turn on Wnt-dependent gene expression. So we looked at this and said, this is a native protein. It's a negative regulator. If we inhibit it, we will increase beta-catenin protein levels. And that's the work we've done. We published this paper in 2024. We made a CTNNB1 germline heterozygous mouse model. We showed it shows the key features of CTNNB1 syndrome in people. We showed that the small molecule, very high selectivity inhibitor of this enzyme is able to normalize beta-catenin protein levels in the mouse, and it's able to improve all of the phenotypes. And we did this treating the mice when they were symptomatic at late adolescent stage. So we didn't even have to start very early in development. And the small molecule was developed by our collaborators, medicinal chemists at the Broad Institute. So I'm going to summarize the main highlights of the paper. Basically, we made the mouse model and we said it's important to prove that it does what we expect. So we found 50% decreases in beta-catenin protein levels in different parts of the brain of the mouse that we sampled, in the spinal cord, and in different skeletal muscles. And these are the tissues that are key to the phenotypes in patients. We've identified novel molecular and functional changes, and these changes impact the excitability of neurons in the brain. And we've found doing behavioral tests that the mice show reduced learning, associative spatial learning. They have impaired motor sequence learning, reduced coordination, and there's more muscle weakness. When we treat the mice with the GSK3 inhibitor, we actually normalize beta-catenin protein levels. So if you look at these graphs here, the blue is always a wild-type littermate. We compare our heterozygous mice in red to the wild-type littermates. Typically, beta-catenin protein levels are reduced in half. If we treat the mouse with the GSK3 inhibitor in gray, you can see the protein levels for beta-catenin now match the wild-type levels. We've shown this in different parts of the brain, and we've shown this in skeletal muscle. We've also shown, I'm not going through all the data, that it will significantly improve the molecular and functional changes that we found in the heterozygote mouse. And the important things we think really are to focus on the learning and the motor skills. And again, if you look at the drug-treated gray, the mice perform in learning tasks almost identical to the wild-type littermates, certainly significantly improved over the vehicle-treated heterozygote mice. So that's in the learning task. They have greater muscle strength as grip strength test, and they have better coordination and motor learning in a different rotarod test. So this is treating them, as I said, at symptomatic stages when they're late adolescence. So what are the take-home features from our mouse work? We developed this model that shows key features of CTNNB1 syndrome in humans. We've identified an efficacious therapeutic treatment, this small molecule, high specificity GSK3 inhibitor. It significantly improved learning and motor capabilities when the mice were fully symptomatic. It normalized the core molecular pathology. It brought the beta-catenin levels up to your normal baseline in different cell types and tissue types. It corrected the molecular, functional, and behavioral phenotypes in the mouse. And we took wild-type mice and treated them with the same drug for the same length of time and saw no significant change in learning or beta-catenin levels, which suggests the first indication that there are no adverse effects. So we're certainly doing a deeper dive into the efficacy and the safety, but this is all mouse. And the important thing is to correct it in humans. So as I said, we've worked with different human cell models, but the one I'm most excited about is our most recent studies working with CTNNB1 syndrome patient-derived cells. And so families like yours have donated blood to Simon Searchlight in the US, and Simon Searchlight is then taking those blood cells, isolating the mononuclear cells, and getting them de-differentiated to a very immature state called a pluripotent stem cell. And they make these cells available to researchers. You apply with a research project. So we have gotten 6 different lines that are available through Simon's that have either a frame shift or a nonsense mutation. These are the most common mutation types that are seen in individuals with CTNNB1 syndrome. We've got these 6 different lines. We've measured beta-catenin protein levels, and they are reduced about 50% in the iPSCs, in these de-differentiated stem cells. But now we want to get closer to a cell type of interest, which is cortical glutamatergic neurons. We differentiated these pluripotent stem cells into the major excitatory neurons in the human brain, in the cortex and hippocampus. And we find that the beta-catenin protein levels are significantly reduced, again, about 50% in the patient-derived neurons. And we've started to look at downstream functions of beta-catenin at the Wnt target gene expression levels, and we're finding decreases there as well. So the important thing is our drug treatment going to show corrective effects in the human cells, similar to what we saw in the mouse. So right now we're working with 2 different GSK3 inhibitors. One is a variant of the one that we showed that works beautifully in the mouse, and that one is 10-fold more potent, so we can use it at greater than 10-fold lower concentration, lower dose. It's always more optimal to keep the dose as low as possible. In our 6 different patient lines, what we're finding as treatment with these drugs in these human cortical neurons will increase the beta-catenin levels. So we're very excited about this result. We took an unaffected parent cell line, treated it with the same drug for the same length of time, so no significant change in beta-catenin protein levels. And we're starting to now look also at downstream changes like the beta-catenin function in the Wnt pathway, and our preliminary data on the subset so far that we've been able to look at have increases in Wnt gene expression. So another thing we're doing that is very new in the lab, and I think is going to be really valuable, is you want to have a non-invasive assay, an easy way to say, is an individual going to respond to the drug treatment, and what is the level of beta-catenin? Because we don't want it too high, that increases your cancer risk. You don't want it too low, because that also, in both cases, too high or too low will cause cognitive deficits in individuals, as has been seen by different mutations and from mouse studies that we've done throughout the years. So we are now taking blood cells, and we're doing a pilot with the mouse. We've taken monocytes from the mouse blood, shown that the beta-catenin protein levels are reduced about 50% in the blood. And if we treat them with the drug 1 or drug 2, drug 1 is the drug we used in the mouse, different doses, we are actually getting significant increases in beta-catenin levels. We think this is a great way to assay whether or not an individual will respond to the drug treatment, and also a way to monitor the level of response so we can always change the dose or frequency. So we think this is going to be a really great assay for us. So we've conducted preclinical studies in the mouse and in human that are developing efficacious and safe small molecule drug candidate. We're going further into the safety aspects of it. What I want to do in the last slide is tell you about another therapeutic approach that was mentioned a little bit by Anna. This is a collaboration we have with Jeff Collar, who's at Johns Hopkins University. So Jeff is an expert in RNA and developing RNA therapeutics. He's very interested in haploinsufficiency disorders like CTNNB1, where you basically have one allele that's functional and the amount of protein that's made is not sufficient in order to really have normal abilities or standard function of that particular product. And so it's based on antisense oligos, but it's a modified approach. It's very clever. So you take this little guide RNA and you design it to recognize the mRNA from the wild type allele, the wild type CTNNB1. It's very specific. It will only find that mRNA in the cytoplasm of a cell. It will bind to this untranslated region, and it has attached to it a poly-A tail. Now, poly-As are attached to all of your mRNAs, and they control how long that mRNA is around. As they get degraded, the mRNA goes away, it's degraded. You don't want it expressed continuously. That would be too much protein synthesis and has its own problems and creates problems for the cell. But the poly-A tail now is added with this little guide. So it's an extra poly-A tail that keeps this mRNA around a little bit longer, and it attracts proteins that increase translation. So from the wild type mRNA for CTNNB1, you get more beta-catenin made in the cell. And we've been testing this with the constructs that Jeff has made, and we found that in different human-derived neurons, and most recently I'm showing you here 2 different patient cell lines, that we can actually increase the amount of beta-catenin in patient-derived neurons with this, what he calls the polyamimetic or the booster. And so this is another therapeutic approach that we're very excited about and think it has great promise. And I'm happy to end there and thank you all. I know I'm the last talk of the day, so I hope you've enjoyed the conference.